We have taken cells from the carcass of an animal that died decades ago and brought them back to life.
Imagine a world where dinosaurs walk the earth again, or extinct animals come back to life. Scientists are working in secret labs, using cutting-edge technology to bring these creatures back, and the results are mind-blowing. Dinosaur cloning. Cloning dinosaurs has fascinated people for years, thanks to movies like Jurassic Park. The idea of bringing these prehistoric creatures back to life feels both exciting and a little terrifying. Let's break it down and see if it's possible. First, cloning requires deoxyribonucleic acid, which is the genetic blueprint for life. For dinosaurs, this deoxyribonucleic acid would need to be preserved in something like ancient bones, teeth, or even a mosquito trapped in amber after drinking dinosaur blood. Sounds cool. Unfortunately, deoxyribonucleic acid doesn't last forever. It breaks down over time, especially over millions of years. So far, scientists haven't found any usable dinosaur deoxyribonucleic acid. Even if we somehow found tiny fragments, piecing together an entire dinosaur genome, all its deoxyribonucleic acid, would be like trying to finish a massive jigsaw puzzle with most missing pieces. And the idea of filling in those gaps with deoxyribonucleic acid from frogs, as seen in Jurassic Park, wouldn't work. Frogs aren't closely related to dinosaurs. Birds or crocodiles would make more sense. But we can't recreate a dinosaur without the full deoxyribonucleic acid blueprint. Another hurdle is the cloning process itself. Modern cloning relies on transferring deoxyribonucleic acid into a living egg cell of the same species. Since dinosaurs and their eggs are long gone, this step cannot be completed. The bottom line is that closing dinosaurs, as we imagine, is just not possible with today's science. While cloning isn't on the table, we've still made incredible discoveries about dinosaurs. China, in particular, has uncovered some amazing fossils, including eggs with well-preserved embryos inside. One of the most famous finds is called baby Yingliang, an embryo at least 66 million years old. This tiny dinosaur, believed to be an oviraptorosaur, was curled inside its egg in a position similar to how modern bird embryos prepare to hatch. This posture, called tucking, helps birds break out of their eggshells. It's fascinating to think that some dinosaurs may have had behaviors similar to birds, their modern relatives. Other discoveries in China include the smallest known dinosaur eggs and even crystallized egg fossils. These help scientists study how dinosaurs laid eggs and lived in their ancient environments, giving us a clearer picture of life during the dinosaur age. Even though we've learned so much about dinosaurs from fossils, cloning them remains impossible. Deoxyribonucleic acid doesn't survive long enough for us to use it. The oldest deoxyribonucleic acid found is about 1 million years old. But dinosaurs went extinct around 66 million years ago. That's a massive gap. Even with breakthroughs in genetic engineering, creating a real dinosaur isn't something we'll see anytime soon. Some scientists have explored reverse engineering modern birds, like chickens, to bring out dinosaur-like traits such as tails or teeth. But even if successful, these wouldn't be actual dinosaurs, just modified birds. For now, dinosaurs will stay in the past. But their fossils continue to teach us incredible things about their world. Who knows what discoveries the future holds? While cloning may be science fiction, the science behind these ancient creatures is as real and thrilling as it gets. If you're fascinated by bringing extinct animals back to life, hit that like button and subscribe for more amazing stories. You won't want to miss what's coming next. Pyrenean ibex, bucardo, cloning. Imagine bringing an extinct animal back to life. It sounds like something straight out of a sci-fi movie. But in 2003, scientists in Spain made history by cloning a Pyrenean ibex, also known as the bucardo, a species that had gone extinct just 3 years earlier. While the experiment was groundbreaking, the clone survived only a few minutes due to a lung defect. The process began with skin samples taken from the last known bucardo in 1999, just before the species went extinct. Researchers extracted deoxyribonucleic acid from these samples and inserted it into goat eggs, with their original deoxyribonucleic acid removed. These eggs were then implanted into surrogate mothers, other subspecies of Spanish ibex, and goat hybrids. Cloning is notoriously difficult, and this case was no exception. Only 7 pregnancies occurred out of 208 embryos implanted, and just one clone was carried to term. The newborn Bucardo's death shortly after birth was disappointing but not unexpected, as cloning often leads to developmental problems. Even though cloning copies deoxyribonucleic acid, the process isn't perfect. Transferring deoxyribonucleic acid between cells can cause developmental issues, such as lung abnormalities in the cloned Bucardo. These problems are common in cloning, making it a challenging and unpredictable science. Despite these setbacks, the achievement was remarkable. The Bucardo became the first extinct species to be cloned, even if only briefly. In a way, it's also the only species to go extinct twice. While the Bucardo experiment was a scientific milestone, it highlights why cloning alone can't bring back extinct species or save endangered ones. Cloning is a long, complicated process with very low success rates. Even if scientists create a few healthy animals, a population with limited genetic diversity would struggle to survive. Inbreeding would make them vulnerable to diseases and environmental changes. Experts also emphasize that preserving habitats and supporting natural breeding programs are more effective conservation strategies. Traditional conservation efforts have saved species like the black-footed ferret and the giant panda. The cloning of the Bucardo was a bold and fascinating attempt at resurrection. While it didn't lead to the species' return, it opened the door to new possibilities in science. However, cloning is still out of reach for long-extinct animals like dinosaurs or woolly mammoths. The lack of usable deoxyribonucleic acid and appropriate surrogate mothers makes such endeavors impossible for now. Instead of focusing on returning to the past, scientists are working to protect the species we still have today. And in doing so, they're giving future generations a chance to share the planet with some of nature's most extraordinary creatures. Woolly mammoth. Bringing back the woolly mammoth might sound like something from a movie, but scientists are working on it. Cloning isn't an option since no viable mammoth cells have been found. Their deoxyribonucleic acid has deteriorated over thousands of years. However, researchers are using advanced genetic tools to create a mammoth-like species by modifying the deoxyribonucleic acid of elephants, their closest living relatives. The biotech company Colossal is leading this effort, aiming to reintroduce a mammoth-inspired animal to the Arctic by 2027. Their approach involves editing Asian elephant stem cells with genes from frozen mammoth specimens. Using tools like CRISPR-Cas9, they add traits like thick fur and fat layers to help these mammoplasty thrive in cold climates. The idea isn't to recreate an exact mammoth, but to blend its key characteristics with those of elephants, even making them resistant to diseases that currently threaten elephants. This work has already reached a milestone. Scientists have successfully created stem cells for Asian elephants, a major step toward producing mammoth-like animals. These cells can also help conserve endangered elephants by offering insights into their biology, reproduction, and health. Colossal's researchers hope this project will expand elephant habitats and combat climate change by restoring Arctic ecosystems, which could reduce carbon emissions. Still, not everyone supports the idea. Critics argue that resources should focus on saving endangered species alive today, rather than trying to revive extinct ones. Others worry about the practicalities. Would these hybrid animals survive in the wild, or face extinction again? Meanwhile, Colossal remains optimistic, emphasizing the potential benefits of its work for conservation and science. It envisions herds of socialized mammophants thriving together, not just a lonely zoo attraction. On a lighter note, another quirky project has involved mammoth deoxyribonucleic acid. Scientists in Australia used it to create a meatball by combining mammoth protein with African elephant deoxyribonucleic acid. The lab-grown meatball isn't for eating. It was more of a science experiment to spark conversations about sustainable food. And apparently, it smells unlike anything we've experienced in thousands of years. While the mammoth may not walk the earth yet, these experiments show how science pushes boundaries, blending the past and future in fascinating ways. Tasmanian tiger cloning. The thylacine, often called the Tasmanian tiger for the stripes along its back, was a carnivorous marsupial once found in Tasmania. It went extinct in 1936 when the last known thylacine died in a zoo. For decades before that, European settlers in Tasmania wrongly blamed the animals for killing their livestock. As a result, Thousands were hunted and killed, and their skins were exchanged for government bounties. Scientists are working on a bold project to bring the thylacine back to life. Researchers aim to recreate a version of this lost species using stem cells and gene editing technology. Australian and United States teams, including those from Colossal Biosciences and the University of Melbourne, are leading the effort. Professor Andrew Pask, who has sequenced most of the thylacine's genome, believes the first baby thylacine could be born in about 10 years. They plan to use stem cells from a closely related marsupial, modifying the deoxyribonucleic acid to match the thylacine as closely as possible. Reviving the thylacine could help restore Tasmania's ecosystem. When the species disappeared, the island lost its top predator, which controlled herbivore populations like wallabies and kangaroos. Scientists hope reintroducing the thylacine could bring balance back to the forests. This project has gained momentum thanks to significant funding and new technologies. Colossal Biosciences, known for its work on reviving the woolly mammoth, is providing resources, equipment, and a dedicated lab for thylacine research. The project still faces major scientific challenges, but the researchers remain optimistic about making history with the first successful de-extinction. Not everyone is convinced, though. Some scientists are skeptical, calling de-extinction fairy tale science. Critics argue the focus should be on conserving animals alive today rather than attempting to revive extinct ones. Despite the doubts, the team behind the thylacine project believes their work could have far-reaching benefits, not just for the thylacine, but for advancing science and conservation. If successful, this would mark an extraordinary achievement, bringing back to life a species thought lost forever, walking through Tasmania's forests again. Quagga de-extinction. In South Africa, conservationists are trying to bring back the quagga, a unique zebra with unusual striping patterns. But there's a catch. The quagga has been extinct since 1883. Instead of using cloning techniques, the team applies selective breeding using zebras with similar characteristics to the quagga. The idea is to breed zebras that look more like quaggas, but there's some debate about whether this effort is worth the resources. The quagga's history is tied to European settlers in southern Africa, who saw these animals as competition for their livestock. This led to ruthless hunting, and the quagga was driven to extinction over time. The last known quagga died in Amsterdam in 1883, but no one realized it at the time, and it wasn't until years later that people understood the species was gone for good. Some surprising deoxyribonucleic acid evidence makes the quagga's return possible today. In 1984, scientists analyzed the deoxyribonucleic acid of quagga skins and discovered that the quagga wasn't a separate species, as once thought, but a subspecies of the plain zebra. This new information sparked a question. Could plain zebras be bred to resemble quaggas? Since zebras and quaggas are closely related, some scientists began working on a project to breed zebras with quagga-like features selectively. The Quagga Project, launched in 1987, has been working on this goal for decades. The project selects zebras with traits similar to the quagga, like fewer stripes and a darker browner coat, and breeds them to create animals that resemble the extinct species. These animals now roam in several national parks and private reserves in South Africa. However, the idea of bringing the quagga back has raised questions. Some critics argue that this is a waste of conservation resources when so many other species are in danger. Others worry that even if these zebras look like quaggas, they may not behave similarly. Still, supporters believe that recreating a version of the quagga could help restore the area's natural history and inspire others to care about wildlife conservation. The quagga we see today might not be the same as the one that roamed southern Africa, but that doesn't mean it's not an important step. Like other animals that were once near extinction, like the bontebok or black wildebeest, conservation efforts can sometimes bring surprising results. In the case of the quagga, it is a new chapter in conservation history. Przewalski's horse cloning. Scientists are celebrating the birth of the second cloned Przewalski's horse, born on February 17th to a domestic surrogate mare. This foal is a genetic twin of Kurt, the first cloned Przewalski's horse, born in 2020. Both are clones of a male Przewalski's horse stallion, whose cells were preserved over 40 years ago at the San Diego Wildlife Alliance's Frozen Zoo. The birth marks a major achievement in efforts to help protect and increase the genetic diversity of this endangered species. Once extinct in the wild, Przewalski's horses have been living in zoos for the past 40 years. Most horses alive today are descended from just 12 individuals, and conservationists have been working hard to introduce them back into their natural habitats in China and Mongolia. However, more genetic diversity is needed to ensure the species' long-term survival, and cloning is seen as a way to help. This new foal is a sign that cloning could become a useful tool for conservation. Cloning, which had only produced a single cloned endangered animal until now, is showing promise as a way to revive lost genetic diversity in endangered species. Experts believe this could help populations grow stronger and more resilient, giving species a better chance of survival in the wild. The foal, described as healthy and active, will soon be moved from the cloning facility in Texas to the San Diego Zoo Safari Park. There, he will join the herd of Przewalski's horses. Though, since he wasn't born into a wild herd, experts will help him learn how to interact with the others. Kurt, the first cloned Przewalski's horse, is already getting the hang of his wild horse behavior and will soon be joined by the new foal. The ability to clone animals like the Przewalski's horse offers hope for species facing extinction. Using cloning to restore genetic diversity, scientists can strengthen efforts to protect and grow populations, helping ensure these animals thrive in the wild for years. Monkey cloning. Chinese scientists have successfully cloned the first rhesus monkey, a species widely used in medical research because its biology is similar to humans. This breakthrough could speed up drug testing, as cloning allows researchers to create genetically identical animals, leading to more reliable and consistent trial results. Cloning, a process where scientists create an identical copy of an animal, is not new. The most famous cloned animal is Dolly the sheep, created in 1996. However, cloning monkeys has been much more difficult. Previous attempts with rhesus monkeys either failed to produce live births or resulted in babies that died shortly after birth. The Chinese team's new cloning method has proven successful. They say the cloned monkey has stayed healthy for over 2 years, which shows that their technique works. They used a method similar to Dolly's. They took cells from one rhesus monkey and reprogrammed them into embryos. These embryos were then implanted into a surrogate mother monkey. However, this achievement has raised concerns. Some animal welfare groups, like the Royal Society for the Prevention of Cruelty to Animals, have expressed worries about the suffering that cloning causes the animals. They believe the pain and stress the monkeys go through are too great, and the benefits to humans may not justify it. Rhesus monkeys are used in medical experiments to study infections and immunity because their genetic makeup is close to ours. Cloning them could make experiments more precise, but the process is still challenging. For example, in earlier cloning attempts, the embryos often failed because the placentas, which nourish the developing baby, didn't form properly. The researchers overcame this by using a special technique where they removed the part of the embryo that would have turned into the placenta and inserted it into another embryo that could develop a healthy placenta. This breakthrough follows the cloning of macaque monkeys in 2018 involving cloning techniques. While those monkeys were important, rhesus monkeys are preferred in medical research because of their closer genetic relation to humans. This success may open the door for more cloning of primates, but it also raises important questions about ethics and animal welfare. Passenger pigeon de-extinction. In 2012, scientists began an exciting project to bring back the passenger pigeon, a bird that once filled the skies of North America in huge flocks, but went extinct in the early 1900s. The idea behind the project is to use modern technology, like cloning and genetic engineering, to revive the species. The goal is not only to bring back the pigeon, but also to help restore the health of the forests in the eastern United States, where these birds once played a crucial role. The passenger pigeon was once the most abundant bird in North America, with billions flying across the sky. However, with the rise of new technologies in the 1800s, such as the telegraph and trains, hunters could quickly find and slaughter massive numbers of pigeons to sell them in markets. This led to their extinction by the early 20th century, with the last known passenger pigeon, Martha, dying in 1914. Now, scientists are using the deoxyribonucleic acid of preserved pigeons to try and recreate a bird that looks and behaves like the passenger pigeon. The closest relative to the passenger pigeon is the band-tailed pigeon, and researchers are working on altering its deoxyribonucleic acid to include traits that make the passenger pigeon unique. For example, they hope to recreate its social behavior and distinctive features like red breasts and wedge-shaped tails. Once the deoxyribonucleic acid is ready, they plan to produce embryos in band-tailed pigeons, which could eventually hatch into passenger pigeons, or birds similar to them. The hope is that restoring this bird will help revive the forests of the eastern United States. Historically, passenger pigeons helped create a natural cycle in the forests by causing disturbances as they flew in large flocks, making room for new plants and trees to grow. These disturbances helped many species of plants and animals thrive. Today, without these natural disturbances, some species are struggling. Scientists hope to restore this natural balance by bringing back the passenger pigeon and creating healthier, more diverse forests. While this project is groundbreaking, there are still many challenges. The team has much to learn about the pigeon's social behavior and how they lived in the wild. Even if they create a passenger pigeon, it will still take time to reintroduce the species successfully into the wild. Nonetheless, the project offers a hopeful glimpse into the future of conservation and de-extinction. Bison de-extinction. The American bison, often called buffalo, symbolizes North America. At one point, millions of these majestic creatures roamed the Great Plains from Canada to Mexico. However, by the late 1800s, their numbers had dropped to only about 1,000. Overhunting, harsh United States, military policies, and westward expansion contributed to their near extinction. The bison had been essential to the Plains tribes, providing food, clothing, tools, and shelter. It was crucial to their way of life, which made the U.S. government's effort to eliminate the bison even more devastating. As the government pushed native tribes off their lands, they also hunted the bison to strip indigenous peoples of their resources and force them onto reservations. By the late 1800s, nearly all bison had been wiped out, and native people were confined to reservations. The United States government, which had once driven the bison to extinction, later set up national parks like Yellowstone, where the few remaining bison were protected. In 1894, the United States Army, stationed in Yellowstone, captured a poacher named Ed Howell for killing bison illegally, marking the beginning of federal wildlife protection laws. The Army was then seen as the protector of wildlife, even though they had been responsible for the bison's near disappearance in the first place. Despite these efforts, the bison population was nearly gone, and it wasn't until the 20th century that conservationists began working to bring the species back. There are more bison in the wild today, though their numbers are still far from what they once were. Now, a Texas biotech company called Colossal is collaborating with indigenous communities to help restore bison to their native lands. They hope to use advanced genetic technology to overcome challenges like the disease brucellosis, which affects some of the remaining bison populations. Interestingly, scientists in Russia have also discovered a well-preserved, extinct bison specimen from thousands of years ago. They hope that this could be a breakthrough for cloning ancient bison. While the chances of success are uncertain, researchers are excited about the possibility of reviving this lost species and reintroducing it to the wild. The story of the bison is not just about conservation. It's about reclaiming an iconic animal central to indigenous cultures and North America's natural landscape. Dodo resurrection. The dodo, a strange flightless bird from the island of Mauritius, is often thought of as the ultimate symbol of extinction. It was around in the 17th century, but when sailors arrived, they brought invasive animals like rats and began hunting the dodo. Since the bird had no natural fear of humans, it was wiped out in just a few decades. A team of scientists is working on an ambitious project to bring the dodo back to life using cutting-edge technology. By studying ancient deoxyribonucleic acid, gene editing, and synthetic biology, they hope to open new doors for bird conservation. Beth Shapiro, a professor at the University of California, is leading the project. She's already sequenced the dodo's genome using deoxyribonucleic acid from dodo remains found in Denmark. The next step is comparing this genetic material to the dodo's closest relatives, like the Nicobar pigeon, to determine what makes a dodo a dodo. However, reviving the dodo won't be easy. The team plans to use a technique involving primordial germ cells forming sperm and eggs. They'll edit these cells with the dodo's deoxyribonucleic acid and inject them back into an egg at the right stage of development. Even if they succeed, the dodo that results won't be a perfect clone. It'll likely be a hybrid version of the bird. Shapiro believes that mastering this technology could be a game changer for bird conservation. By transferring helpful genetic traits between bird species, scientists could protect them from diseases and help them adapt to a changing world. But the journey to bring the dodo back is filled with challenges. Experts like Mike McGrew, a lecturer at the University of Edinburgh, say the difficulty lies in adapting this technique for other bird species. not just chickens. The project has attracted considerable attention, including investment. Colossal Biosciences, the company behind the effort, has raised millions of dollars. However, some critics argue that the money could be better spent protecting endangered species that are still alive. Despite the debates, the dodo's story fascinates people and even appears in books like Alice in Wonderland. While much about the dodo remains a mystery, it forever changed how we think about extinction, symbolizing both the beauty and the tragedy of lost species. The idea of cloning dinosaurs and bringing back extinct animals might sound like something straight out of science fiction, but scientists are making real attempts to bring the past back to life. While there are still many challenges ahead, the advancements in genetic technology show that nothing is truly impossible. As these experiments continue, we may see the impossible become a reality, raising exciting possibilities for science and conservation. Only time will tell if these daring efforts will succeed or not.